Good evening, everyone. Thank you for joining to this wonderful session called Gene Drives, Nagoya Protocol, and Digital Sequence Information. These terms might be very new for some of you, but they are very fascinating. Well, I have to take you back almost to the end of the 19th century when one of the greatest scientists of all time, Gregor Johann Mendel, experimented at his backyard in his garden, where he transferred the pollen grains from anthers to the stigma. And what he did was he transferred the pollen grains from a tall pea plant to a short pea plant. And he waited for the result. The result was so amazing that both the tall and short plant gave rise to a hybrid plant. Though the characters of that plant was tall plant, so he come to a conclusion that there are some genes uh, within that organism that are so dominant. So like that, he started to continue to transfer the pollen grains and continued his experiment. And that's how we understood the importance of genetics. And the basic principle of genetics is inheritance and variation. Means the characters from your parents can be transferred to you. At the same time, there will be variations within you. So what are all these things that I am referring to? And what are these variations? What are these inheritance? What are genes, etc.? I welcome you to the world of genetics and biology. Before I give the floor to our wonderful speaker, we have a poll questions ready for you all. I request all of you to participate in that and see uh, how you answer them. So now I will share the AHA slide link. And let me share first. In the chat box, I'm sharing the AHA slide link for you. And I will share the screen. And so how well you are versed in biology? I am passionate about biology and want a career in it. I like biology, but we are getting responses. Yeah, some say they are passionate, they want a career in it. Some says I like biology, but I like different subject. On a scale of 10, yeah, we see some more responses. I like to know about biology, but sometimes it's difficult. And this is like 6.8. That's fine. I mean, it happens. And we see at 2.6, I like biology, but I'm passionate about other subject. Well, I think we are getting more feedback. Not much interested, but I just like some of them are saying. I think the average for the blue one is somewhere around 5, which is not good. That is, I'm passionate about biology and I want a career in it. And some say I like biology. The average is 5. Okay. You can see the blue one average is 2.3, which means that very few people are in this forum where they like biology and they want a career in it. Let's see, 3.6, not much interested, but yeah, I just want to know the basics. And the green one, I like to know about biology, but it's become a little bit difficult for them to understand. So I think no need to worry. This session is not about giving you so much of knowledge, but to make you understand and about certain topics which are quite relevant. I see more response. That's great. I think we'll move to the next question now. The next question is, a gene is a segment of DNA and RNA. Select true or false. Your first option is true. Second is false. No, it's only a segment of DNA. Your third option. Okay. That's great. Okay. 60% of them say true. We have 50% on both the sides. Great. 55 is 44. Okay. There you go. I'm glad. Oh, we have false also. Okay. 45.5, 45.5. It means that 5 people said true, 5 people said false, and 1 said. Okay, we got one more response. Yeah, kindly fill this. We'll understand like how. Okay, we are getting more responses, more responses. You can see the response. Yeah, we have 15 responses and 60% of them say true. Okay, we are getting more responses. Great. 37.5% says it's only a segment of DNA. That's great. I can give you a hint. Think about virus. If anyone didn't answer it, they can actually now do it. Think about virus and answer this question. Okay, I think, yeah, we are getting good responses, 58.8. So I will show the correct answer to you. The correct answer is option A. Yes, true. A gene is a segment of DNA and RNA. A gene in viruses, the genetic material is generally, I mean, it's DNA and RNA, but a gene can be a segment of RNA also. So I hope that you understood what is gene here and how it is made of. Let me go to the next question. Where do you find the DNA in a cell? Is it chromosome? Is it lysosome? Is it ribosome? Or is it? Wow. We got 2 responses. We got 3. Chromosome. I'm glad that you know. Any more answers? Think about microbodies also before like answering this. Think about ribosomes also. Are there any DNA in ribosomes? We got 12 responses. Everyone is saying chromosome. So I think I'm gonna share the answer to it. The correct answer is yes. Oh, we have some response even for microbodies also. But that's fine. This is generally for your awareness that DNA is part of chromosome. So let's go to the next question. That is, pick the structure of chromosome from the below image. There you go. You have 3 structures. There you go. Some people pick the option B. I don't know which is the answer here. Look carefully at the images and select your answer. Carefully look, try to recollect your basics. Come on. Okay, we have some responses for all the 3 options. We have A, okay, 10%, B, C, 10%, and but we have B like almost 81.82%. Okay. Some people are picking the option C. Okay, great. Come on, guys. Yeah, I can see more response. Some people are picking up this option C, which is good. Yeah. I'm not seeing anything about option A. I think, don't you think it has anything to do with the structure of chromosome? Check carefully. I will wait for another 10 seconds and I will show the correct answer. Well, thank you so much for this. The correct answer is option B. Yes, that's the structure of chromosome. As you can see, the DNA helical structure coming out of chromosome. So DNA is part of chromosome and gene is a segment of DNA. I hope you understood this concept now. So let's go to the next question. What are amino acids? Are they fats? Are they carbohydrates? Are they proteins? Or they are nothing but ammonia plus hydrochloric acid? Wow, we got 2 responses. Wow, we got 3. One says we have 3 options. Great. What about option D? Don't you think amino acids are nothing but an acid and a base together and they become amino acid? Okay, we got a good response for option C. Okay, we got 1 good response for option D, which is good. Option A and option D are at low. Many people are responding to option C. Some are responding to the third one. Oh, fourth one. Okay, that's great. That they think that amino acids are nothing but ammonia plus hydrochloric acid. I mean, I love your interpretation. But we are not seeing any response for option A and option B. I wait for 10 seconds and I will show you the answer. Amino acids are the building blocks of life. So now you have seen that chromosomes are part of nucleus and DNA is part of chromosome and gene is a segment of DNA, and a gene actually contains several codes, and these codes are nothing but amino acids. That's why they are called as the building blocks of life. I mean, if you are confused, no need to worry. We will explain it again. This AHA slide is just to make you understand. The correct answer is proteins. Amino acids are nothing but the proteins. So let's go to the next question. Okay, when I say adenine, guanine, thymine, cytosine, and uracil, pick the correct one from the below. They are nothing but nucleotide base pairs of DNA and RNA. They are different types of fats. They are solvents. They are pharma drugs. Pick your answer. Let's see. We got 3 responses, 4 responses, 5 responses for option A. Okay. We have 6 responses for option A. Okay, great. We have 11 responses for option A. They think they are nucleotide base pairs of DNA and RNA. Think we also have other options. Think. Do not just quickly admit your answers. We have 16 responses for option A, 17, 18 for option A. I am not seeing anyone picking up other options. So this is great. This is simple. I think you understood. Yes, when I say adenine, guanine, thymine, cytosine, and uracil, they are nothing but the nucleotide base pairs of DNA and RNA. So in a DNA, you have adenine, guanine, thymine, and cytosine. Whereas in RNA, you have adenine, guanine, cytosine, and uracil. So that's the basic difference, but they are nucleotide base pairs. So let's go to the next question. Well, we all are hearing about vaccination. The antibiotic penicillin is obtained from, okay, some says fungus. I got the response. You also have bacteria. Remember Alexander Fleming? Okay, I see someone picking up the option D. It's nothing but virus infected cells. Oh, great. We are getting response for option A, and there is a battle between 3 options. I see option B going down, and many people are responding to option A. I think that's great that you remember about Alexander Fleming. I see some responses for option B. but some also say it's option C, by synthetic means. Okay. I wait for 5-10 seconds. So Alexander Fleming did his experiment on a bacterium called Penicillium, and he observed that in a lab, a fungus was growing on this bacterium. Sorry, I don't remember the bacterium name, but he observed that on the bacterium, a fungus was growing, and this fungus was inhibiting the growth of the bacteria, and he called this fungus as antibiotic. And the correct answer is fungus. I hope that you understood. Now let's go to the next question. Well, this will be the last question for now. Which is the first genetically modified crop in India? I think it could be a little bit difficult, but you can try your best. Oh, wow, I see 4 responses for option B, some response for option C, and some are for, I think, great, 11 responses. I see some for A, some for C. Okay, I see option B, very stable. Yes, ladies and gentlemen, I think I will show you the correct answer quickly. I'm getting one more response. Okay, I will wait for 5 seconds. I will wait for some more time. Let's see which answer you will pick. Okay, the first genetically modified crop in India is BT cotton. Well, BT brinjal is still in stages. I mean, there is no BT rice till now. Okay, thank you so much for participating in this session, in this poll, and I hope that you understood a bit of it. If you understood anything about DNA gene right now because of these questions, just type yes. If you didn't understand, type no. The option is yours in the chat box. All your questions will be addressed at the panel discussion. Right now, keep your questions and maybe write down your questions somewhere and ask at the end. Now, I take this pleasure to invite Dr. Ankur Sharatkar all the way from University of California, San Diego. It's early morning for him. Good morning, Ankush. And he completed his master's from Velour Institute of Technology, and then after that he did his PhD from University of France. Then he defended his thesis over there, and he studied about the protein molecules and the plasma membranes in Drosophila, and he did so many experiments on genetics, and right now his experiment is mainly on gene drives. So I hope that you understood a bit of genes. So Ankush will explain to you what are these gene drives and whether they are useful or not. And so I just like to give welcome Ankush and ask him to share his experience. And yeah, Ankush, thank you.
Thank you, Bharat, for inviting. So I'll be talking about gene drive. So I will be talking about what are the promises and the side effects of the gene drive, which is a new technology right now. So just to get a brief intro about what is gene drive. So the first thing we have to know is how a gene is inherited in normal population. So this is the gene inheritance in mosquitoes, basically. So here a transgenic mosquito mates with a wild type mosquito, and it undergoes Mendelian inheritance, which is a normal way of inheritance, where like 50% of progenies will get a transgenic element and the other 50% will get wild type gene. So this is how in a normal inheritance is happening, which is a Mendelian fashion, where 50% of progenies will get the transgenic and 50% will get the wild type allele. So, and then in the similar way, as we go to the next generation, it will be again the 50% when it mates with again a wild type mate, there will be 50% of alleles, which will be 50% of transgenic flies, mosquitoes, and 50% will again be wild type. So that's how a normal inheritance of a transgene happens. So if you see in the last generation, like there will be a dilution of the transgenic element, and you will have mostly a wild type population of mosquitoes. And whereas a gene drive element or a transgenic element will be uh, really in a small population. So that is how a normal Mendelian inheritance takes place. But then there is a new synthetic way of inheritance, or a new super inheritance. It's called a super Mendelian inheritance, where a transgenic element gets this transgenic mosquito is called as a selfish gene carrying element, or a gene drive. So where all the progenies after mating with a wild type mosquito, it will give rise to all transgenic mosquitoes. So that is how, and in the end, like if you go down to the next generations, it will just give rise to all the transgenic mosquitoes. So here, this is a selfish gene which will just inherit itself, and it will not allow a wild type gene to propagate in a population. So how does this inheritance comes into picture? So there are many factors which contributes to this super Mendelian inheritance. That is like viral elements. So as most of the organisms on this planet, like they have like some or other viral element in them. So our like humans, they also have like a lot of viral gene embedded into our chromosomes. So virus has a property where it can insert itself into the genome of a species, and thus give rise to a leads to copying of itself. So giving rise to a drive like this, where it is like super Mendelian. Or like chemical agents like DDT. So people have used DDT to control mosquitoes, but what DDT has done, it's like it has the mosquitoes had acquired a particular mutation through which it has gained resistance to this chemical agent. And our use of continuous chemical agent has just given rise to mosquitoes which are carrying that particular mutation, which is giving rise to resistance to DDT. So that is also a way of giving rise to a super Mendelian inheritance. And the new concept is a gene drive, where it's a completely new element where it can just copy itself from one gene to another gene. So I will get into this this topic of gene drive now. So how it is happening? So a gene drive element, here you have one allele from paternal chromosome or a maternal chromosome, and the other from the other parent. So one of the gene will have this specific element which has Cas9 and guide RNA. So Cas9 and guide RNA both will make a scissors to cut the genome, and it can copy itself. So that is how gene drive happens. And this this Cas9 and guide RNA element are from bacteria. So this scissors is made in bacteria to fight against viruses and other elements, so that they can protect itself. And this thing has been engineered in mosquitoes to copy the gene of interest, so that it can be like all the population in the end will have this gene drive throughout the population. So this is like, initially it was developed in fruit flies. So here you have on one gene, you have Cas9 and guide RNA, where it goes and cut the another chromosome. And thus like it will copy this Cas9 and guide RNA whole cassette into the another chromosome, and thus giving rise to this homozygous flies, which are yellow body color. So this was initially done in fruit flies, like just a proof of concept. So this is how like you can convert like heterozygous mutation into homozygous mutation. So by using a gene drive. So what are the applications of this technology is there are two main applications. So one is population replacement, where you slowly introduce this transgenic mosquitoes, which will copy itself throughout the population, And in the end, you will have a population which will be just be this transgenic mosquitoes. So this is one way. And the other is for population suppression. So where like in like a lot of places like US or in Hawaiian places, like where mosquitoes were not present earlier, and they were brought here by because human migration and everything. And in these places, the mosquitoes could be suppressed. So here, like they can be completely removed from the ecosystem. So here these mosquitoes are sterile. So like when they start through the population, they will completely remove the wild type population and just suppress the whole mosquito and remove the whole mosquito from the ecosystem. So these are the two main applications. And how does this two comes into picture? Let's see. So here, like you have this Cas9 guide RNA, which will cut and copy itself. And you have this cargo element, basically. So what happens is, because it is copying itself, like, and if you introduce this particular mosquito into a population, it will slowly drive through and remove all the wild type mosquitoes and convert everything into transgenic. So, and this is like a data which was done in mosquitoes, which shows that like when they introduce this transgenic mosquitoes, it slowly drives by generation 8. You will be completely removing the wild type mosquitoes, and it will be just gene drive mosquitoes in the end. So this is how successful this population replacement can happen. And here you have to do nothing. Like initially in the first generation, you introduce this mosquitoes and it will drive through and remove the whole wild type population. And in the second technology is about like population suppression. So it has been done in mouse. So here what happens is like it's called male germline inheritance, basically where the Y chromosome will have this gene drive and it will just cut the X chromosome so that like only Y chromosome is viable and X chromosome is not viable. So all the progenies you will be getting will be male mouse. And thus like from this cross, like where a male carrying this population suppression gene drive will just give rise to male progenies. And in the end, like there will be a population of just male progenies in the whole population. So which will completely suppress the whole population like this, like because there will be only male mosquitoes coming in, male mosquitoes or male mouse are getting made, and then it will lead to like complete removal of the population, and we can get rid of the mouse population in a particular island or in a given scenario. So these are like main two applications, and where it can be applied is like there is wide area where it can be applied. Like one is like blocking the virus carrying mosquitoes. Like basically we can immunize a lot of animals not to carry harmful diseases. Like there are kissing bugs which can carry Chagas disease, so for which there is no cure. So whether we can use this gene drive element to stop this kissing bugs from carrying the Chagas disease, or in mosquitoes from carrying dengue, malaria. So how does that can happen? So as I have shown you, this gene drive element has the cargo. So in cargo, what we can do is like we can put anti-plasmodium. Plasmodium is a malarial parasite. So we can put genes which can stop a particular parasite like plasmodium, or it can stop viruses like dengue and Zika. And thus like this mosquitoes will not be able to carry the particular pathogen. So we will be getting rid of the whole population carrying this pathogen, and we will get rid of malaria and other diseases. And the other main problem is also like pest, where like rodents have been pesting on agriculture, or in New Zealand they have found that like because of human invasion, the rodents have come into the New Zealand. And as you know, like there are a lot of flightless birds in New Zealand, and rodents, they are like taking over the nest of this flightless bird, and there they want to apply this sterile insect technology, where like population suppression in mouse, so that they can just release this mouse, which will lead to just male progenies, and in given time it will just suppress the whole, remove the whole mouse population from the New Zealand. So that is one of the application where the suppression could be used, and mainly that is one of the application. And it can also be used to welfare of lot of organisms, like reducing starvation in lot of animals. And like, for example, in cheetahs, like here in the cargo, like as you know, in cheetahs the problem has occurred that like because of continuous inbreeding, they have lost their ability to fight against different pathogen. So here in the cargo we can put some specific immunizing factors so that they can fight against different pathogens. So that can happen, and we can save cheetahs from dying, and also other mammals too. So this is one of the application. So this technology has great promises, but there are like a lot of problems too. So one is that like it can change the ecosystem completely. So mosquitoes are like main part of the ecosystem, and there are a lot of like fishes which feed on mosquito larvae. There are birds which feeds on mosquitoes, and they're like, mosquitoes are important pollinators. If you remove mosquitoes or like do a gene drive in mosquitoes, like you will completely change the ecosystem. So that is a cost like which this technology comes with. And the other thing is, as I have shown you, like in fruit flies, they have made a drive initially where the wild type is darker body color, and in the gene drive, it's lighter body color. And you can see like modified flies too, which will have like half and half, so which are resistant to drive. So it gives rise to various mutations in the genome of these flies, so which we have to really be worried about. Like this technology can cause a lot of mutations in particular organism. So that is one of the main problem of this using the transgenic drive. And then as I have told you, like we can use the in cargo different immunizing factors or anti-malarial factors, for which we don't know what the side effect would be. Like maybe the mosquitoes will be like, as we are seeing with different, like the Zika, this SARS virus, it is gaining immunity against like it is rapidly evolving. So here also, like if we use an immunizing factor, it can rapidly evolve to not be immunized against this particular cargo which we are putting. So, and we don't know what will be the effect of that particular immunizing factor. So one thing we have to be like really be worried about. And there are like, there will be a lot of unintended outcomes from this gene drive, like which, because this is a new technology, we don't know what would happen if it is introduced into the wild. And there are like ethical concerns about modified mosquitoes and modified organisms in the population. So this is what the gene drive is about, and I have shown you like what it has promises and what problems it has. Yep. And we can take so that's it. Thank you.
Thank you, Ankush, for that very basic presentation, explaining us in that pictorial presentation, explaining how actually the inheritance generally takes place. So, I mean, generally what happens in the lab is to make you understand, there will be a crossbreeding of generations. So they keep on experimenting from one generation to second generation, third generation, fourth generation, and they check what is the inheritance levels. Inheritance means the transfer of characters. For example, right now, today, you might have some characters of your great grandfather. You never know. You might have, you may be very different from your parents. So according to genetics, there is one more interesting thing. We think that most of our characters, 90% of our characters come from our parents. No, that's not completely true. Actually, most of the characters come from your previous, your previous generation, means
Great
like
grandfather.
from your father's father, father's mother, or mother's father, mother's, grandfather. So some of you might be like very great at painting. Some of you might be very great at singing. Some of you might be very great at like writing. So these traits, that characters that you get, are generally come from your forefathers. So this is the beauty of genes. This is the basic of like how we got evolved. So I think we have to like applaud Sir Johann Gregor Mendel for his wonderful experiment and explaining to us about the concept of genetics. He's the father of genetics. So let's move on to my presentation. And I thanks once again Dr. Ankush for presenting. I request him to like wait because we have some questions for him. As a moderator, I do also have some questions. So now let's move on. I don't have any AHA slide right now, but I just like want to share my screen to you and tell you some stories. Give me a second. Okay, ladies and gentlemen, thank you so much for joining. So my presentation that I'm going to give, I am Bharat. I have completed my master's in pharmaceutical chemistry and specialization in genetic engineering, DNA synthesis, and instrumentation, etc. Well, I think these two words for most of you should be very, very, very new. But don't worry. Nagoya Protocol and digital sequence information. Well, in the chat, you can just type, where is this place called Nagoya? Don't search in Google. Where is this place called Nagoya? I'm not seeing any answers. You can randomly type. Okay. I think I tell you, it's in Japan. Oh, I saw some answer in China. But that's fine. It's in Japan. Yeah, it's in Japan. Yeah, I already told the answer. So Nagoya Protocol and digital sequence information. Well, I can tell what is this Nagoya Protocol. I think most of you have been attending CBD. One of the third principles of CBD is access on benefit sharing. That is, whatever I access from you, from the provider of traditional knowledge, I have to provide certain benefits to the traditional knowledge provider. So the protocol is the third objective of the CBD, which is a legally binding framework. And it has been ratified. It came into force in 2014 because the Nagoya Protocol is important because the way genetic resources are accessed and benefits from their utilizations are shared can create great incentives for biodiversity conservation and its sustainable use, while also contributing to a fair and equitable share. So I mean, I have defined it, but I'm not going to go into the depth of Nagoya Protocol. I will just discuss certain things, but I want to tell you the story of Nagoya Protocol, and then I will talk about digital sequence information. So my first slide. Okay, you can see this slide. A farmer is growing local crop varieties and traditional knowledge, etc. And this was like long ago. And a businessman one day suddenly pops up and he says, hey, I have a deal. I have a deal for you. I want to do business with you. I want your traditional knowledge. I want your crops, and I would like to pay you 100 rupees per kg. Now the farmer agrees to it because 100 rupees is good amount for him. So he sells that particular product to the businessman, and the businessman takes that product to his industry, and where he manufactures, where he produces a wonderful, gorgeous looking product and sells into the market. Now, here is the thing, there is no problem. When there is no regulation, what happens is that he bought the product from the farmer at 100 rupees per kg, but he's selling the product at 1 lakh rupees per kg, 100,000 Indian rupees per kg. So where is this fair and equitable sharing? They have been doing this, they have been exploiting this. So when there is no regulation, then this can happen. You have no idea. So in the market, what is the final product? If I say this has been happening in the past, yes, it happened, but I can't say that it's still happening somewhere. So this is when there is no regulation, when there is no rule, there is no law. But suddenly, one day, Nagoya Protocol came into force in 2014. Now you just look at the picture. When Nagoya Protocol is in force, you can see a businessman, what he has to do. He has to access the genetic resources. Okay, when the Nagoya Protocol is in force, when there is regulation, and then he sells the product, and then whatever the benefits that are arising by selling those products in the market, the fair and equitable sharing of benefits arising out of genetic resources should be shared. So this is in general about Nagoya Protocol. I hope that you understood in a simple way I'm explaining, but there are like so many implications and there are so many complex things involved in it. So this is when there is regulation, there is a control, there is like step-by-step process, there is transparency. Now, but the businessman is worried, because he has to go through so many steps to like capture the biodiversity, to capture the genetic resources. So he calls everyone he knows, he tries to do anything, but the authorities are strict. They say that you take approval from us and follow the rules, or else we are not going to like give you permission or grant permission to access the genetic resources that grows at our backyard. And this says Nagoya Protocol when it is like strongly implemented. So the businessman is worried, so worried. So what he does? Can you guess what he does? Well, this is how it happens. Unethical practices can happen. In my experience, this is one thing that I have seen. So will be Mr. X. We never know who is this Mr. X. We never know like who is actually coming and bribing all these things. So the Mr. X finds the mediator. Mr. X finds the mediator. These mediators are generally either local people or maybe some kind of mafia or smugglers. We don't know, but they operate through these mediators and reach out to the groups at the ground level who are producing the crops, local varieties, or they might have genetic resources. They are conserving it, and this is like instant gratification. When you look at money for your needs, you generally accept the things. Now the thing is that the consequences of instant gratification is that generally either the mediator get punished, or sometimes even the people actually who conserve the biodiversity get punished. And this is in many cases, it's not just in India but around the world we have been seeing. So because this is Mr. X, we don't know, he's like a Zorro, and where he's hiding and what he's doing. And this is one of the unethical practices. But having said that, there are many complications, I said, with when you like try to implement Nagoya Protocol very strictly. So I think this is one of the unethical practices I'm sharing. There are many to make you understand how generally it happens. The classic example that I want to give, the story that I told you till now, this is in the form of an example I'm sharing. Look, what happened with famous Ongole bull case. People who are from Telangana, they know where is Ongole. Ongole is in in Nellore district of Andhra Pradesh, which is on the eastern coast of Andhra Pradesh, towards Bay of Bengal. Well, in 1950s, 1960s, this famous Ongol bull was smuggled by Brazilians. And these people smuggled the Ongol bulls, and they used to, at that time, they used to pay to the farmers around 10,000, 5,000 rupees, because in 1950s, remember, 1,000 rupees is a great amount. So what they did was they took these bulls in the ships and they started to breed them, and they also started to collect the embryo from the Ongol bulls, and they developed their own local Ongol bull varieties. All the Mexican bullfights that you see in the television, all the American Brahman cattle that you see are nothing but they originated from Ongol bulls. Now, interesting thing, as you see in the below image, the Brazilian Ongol bull is sold by Brazilian people, I mean the Brazilian dairy farms, at 3 to 4 crores, which is 300,000 to 400,000 US dollars. And it is still like, till like 2015, 16, this has been the case. Till there is smuggling of Ongol bull, because, and our farmers sell at around 3 to 5 lakh rupees. But most of them don't know that the fondness the American continent have for Ongol bulls. So I think this is one classic example of Ongol bulls. Now, why this is happening? The question you have to ask is why this has happened and why this is happening, and what is being done to conserve or protect Ongol bulls. I mean, I can share some interesting facts. Like this is Deccan Herald reported Brazilians or Brazil on an embryo hunt for Ongol breed. Yes, in 2014 and 15, Brazil applied. Luckily, Nagoya Protocol came into force in 2014. Like whatever story I explained to you before, now Nagoya Protocol, because of it, the authorities took action. So the Brazilians firms applied for the embryo of Ongol bull, but the authorities didn't give permission so quickly. The authorities checked everything, and they kind of saw why this is happening, and they realized that the economic value of Ongol bull in Brazil is so great, and people are so fond of Ongol bulls. This was in 2015. I think they applied for 500,000 embryos of Ongol bulls, and each embryo cost around 5,000 US dollars. So I think, this was in 2015, but you see why we were unable to conserve. When you have such type of attitudes, read the second statement. I don't like to mention the name, but I can say that its former AP Biodiversity Board member secretary, he visited to Brazil, and he discovered that they had improved the Ongol bull variety genetically. And there are more than 1 crore top quality Ongol bulls in Brazil and Mexico. And according to him, there are not more than 1,000 Ongol bull top breeds in India. This is problematic, people. This is the real concern you have to. This is the mindset that our, most of the people they have. When you have such type of mindset, do not enter into the boards. Do not work there. Resign your job and get away. And you see what happened again. The same. These are again from my own state, our own state, AP. A delegation from, I think most of you know about BRICS. BRICS is nothing but Brazil, Russia, India, China, South Africa. Yeah, Nikshit, I will answer to your question at the end, not right now. So BRICS delegation meeting happened and in Brazil. So some of the delegates went from India, that is from Andhra Pradesh, to Brazil. And there the Brazilian firms have shown the top Ongol bull varieties to them. And these delegates who were there from AP, they kind of appreciated them so much, and they were like shaming our own place, shaming that we were unable to conserve. Are how come you will conserve when you have such type of mindset, man? Come on. So this is what the problem is. We are not seeing our potential. We are not seeing the amount of diversity that we have. Come out of that mindset first. Realize your potential. So right now, the good thing happening is that since 2018, there was one such great move happened that the authorities took, that is NBA took, is the Brazilian firms have to pay 1.2 crore for Ongol bull embryos. So, which is like nearly more than 100,000, 200,000 US dollars, nearly 200,000 US dollars. The firms have to pay if they want to collect embryos. One more interesting thing happened, but I will share in my next slide. So when you have such type of mindset, when you have such type of things, why I'm so concerned, why I'm like raising my voice, because of this particular picture. Read the statement yourself and see what happened. This is a picture in Kurnool district in Andhra Pradesh in 2018. Farmers were selling Ongol bull in Kurnool market at a lower price at around 20 to 30,000 rupees because there was a drought, because there was fodder deficit. I mean, when you have such type of mindset, on one side you are praising, some other country because they smuggled your biodiversity and they started to like develop everything, and now they are telling you that, hey, we will help you now, and at the same time you are appreciating them and you're not concerned about your own people, man. This is the problem. See, more than, I mean, this news is like so shocking about Ongol bull that the farmers, they don't know about their own economic thing. They don't know about their own potential because there was drought, there was fodder deficit. Of course, when this type of things are there, they do smuggle. I mean, they do practice unethical things. Like I was sharing, they will sell Ongol bull at like 3 to 4 lakhs, or maybe when such type of demand is there, when there is a lot of Ongol bulls in the market, they have to sell at a very low price. I mean, this is what I'm trying to tell you, that we have to realize the ground realities. We have to understand our own potential first of all, and for that we have to think deeper and understand even the market around us. Only then I think we can conserve biodiversity. So this is a classic example of Ongol bull that I'm sharing. There are many such type of cases. Hope you understood a bit of it. I think yes, when this Ongol bull case is happening after, like, now let's say that CBD is there and you have Nagoya Protocol is in force, and you think we are conserving biodiversity and we are really great. I mean, everything is fine. But what's next for Nagoya Protocol? Here is the thing I want to tell you. In 2014, Nagoya Protocol came into force. But in 2016, immediately next 2 years, some of the most advanced nations in the world who has great technology, much more ahead than us, okay, they have plans for next 30 years. Do we have plans for next 30, 50 years? We don't even have plans for next 5 years. Even we have, are we implementing correctly? So what's next for Nagoya Protocol? Well, everyone is saying from the countries, mega diverse countries, yeah, I think we are doing great, but wait and see what is coming next. Ladies and gentlemen, coming next is digital sequence information. Look at the picture on your left side. How you want to see diversity? You want to see biodiversity like on the screen on your left side, or you want to see biodiversity like on your right side? I think in our slides, you were actually like you answered one question, like adenine, guanine, thymine. I think they are nucleotide base pairs of DNA, as I told you, and even RNA. Everything can come into lab. So how you want to see? The question is like, I want to ask to you, but let me explain what is digital sequence information. Well, It's very simple. As there is a cell, inside a cell, there is a nucleus, and inside a nucleus, there is a chromosome, and DNA is part of chromosome, and gene is a segment of DNA, and a specific gene codes for a protein. And when you take out that code, when you like take out the entire structure, you will get like a sequence. So you can actually map the entire sequence. I will explain in my next slide. What you are trying to do is you are mapping the entire sequence of an organism, genetic sequence, and you want to store digitally. That's why it is called digital sequence information. The information should be available in computers, like in a software, etc., etc. So there are in the world, there are 3 repositories of digital sequence information where all the genetic information is stored. One is in USA, which hasn't ratified the CBD. Second is in Europe. Third is in Japan. So all 3 countries have great technologies related to digital sequence information. Why? You can think about it. And why they want the biodiversity, you can think about it. But I'm going to share my next slide to you to show you something interesting. How many of you have seen Jurassic Park? Please say yes if you have seen, because this is very important. How many of you seen the movie Jurassic Park, especially the 1992 one? Please say yes if you have seen, because what I'm trying to share is very much related to Jurassic Park. If you remember John Hammond, the character, Dr. John Hammond, Steven Spielberg. Yes, I can see. Yes, yes, yes. This famous scene, if you remember, where Dr. Grant is trying to save the kids in the car. Yes, yes, I can see. Yes, 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 yes. Thank you so much, ladies and gentlemen. Well, if I want to explain to you digital sequence information, I have to explain in simple terms. Well, if you have seen the movie Jurassic Park, in the beginning, Dr. Hammond shows a video, and in the video where he shows that, yes, Shoham, I read you. Thank you. Thank you for joining again. Well, if you have seen the movie Jurassic Park, in the beginning, they show a video where millions of years ago, when dinosaurs were ruling this planet, a mosquito comes and bites the dinosaur. One day, the mosquito, when got stored in a tree sack. So the genetic material of that dinosaur is actually present in the mosquito, and the mosquito is in the tree sack. Well, now when paleontologists discovered the mosquito sack, they actually extracted the genetic material of the dinosaur, which was there millions of years ago, and they started to understand where exactly they can fit the genetic material of dinosaur into which organism. They did so many tests. Finally, they found that it was frog, the reptile, in which the DNA, the genetic material gets fit. And then they then after that, they inserted the genetic material of the dinosaur into the frog. And through that process, they did the molecular cloning, and that's how the dinosaurs got like established in the movie Jurassic Park. Again, they recreated it. Well, I explained to you in a very simple language, but there are many things that happen in this process. But if you remember, there is a famous dialogue from the actor in that movie. God creates dinosaurs, God destroys dinosaurs, God creates man, man destroys God, man creates dinosaurs, dinosaurs eats man, woman inherits the earth. This is a dialogue from, this is not my quote, but this is just for to make you understand how exactly everything is happening right now. So I think this is molecular cloning, they call it. We cloned so many animals. But how this generally happen is, look at this picture. In 2010, in the history of mankind, like we did the human genome sequence. It means that your entire genome sequence is ready right now. So if you give me a blood sample, I can actually sequence your entire genome within like 6 hours. And it cost, I think, less than $1,000 or maybe less right now. So what is this genome sequencing I'm talking about? Genome sequencing or mapping the pattern of the basic building block of every living cell. A genome contains all of living beings' genetic material. Simply put, the genome is divided into chromosomes. Chromosomes contains DNA, and DNA genes are segment of DNA. I'm sorry for that particular thing there, but yeah. So if you look at this picture, the top picture, this is what I have been explaining to you. I hope that this picture is stored in your mind right now. Now, what I'm doing with the genome sequence, that is the digital sequence, I'm taking out the DNA. Let's say you have a specific plant in your like backyard or in your like specific area. And what I'm trying to do is I am collecting that plant, taking to my lab, and I'm sequencing the entire plant in my lab and trying to store it somewhere digitally, safely. Why? Why? Because as you have seen with this picture, like to create something like how I want, how I desire, so that digitally when this particular information is available to me, it will contradict the entire agenda of Nagoya Protocol. You have seen what is Nagoya Protocol. In Nagoya Protocol, when there is a force, the institutes, the organizations cannot access your genetic resources. To access them, they have to undergo several steps. But let's say I have that genetic material with me. I don't have to access and again come to you. I have the genetic information with me. Then there is no question of like, benefit sharing. Whatever I am earning, I am earning. I don't bother about you because I have the information. I'm going to give you some examples like how it has been misused, because BSI is like being developed in some of the countries at a rapid rate, and it is being developed, it has been developed, including in India. Now, the concern that we have to understand, yes, this is again a quote from the movie Jurassic Park. If you read, some of the worst things imaginable have been done with the best intentions. We have the best intentions, yeah, for mankind. I want to do something best, but you see again what happens. So this is the thing happening. Like I told you, sequence of a complete human genome like can be done within 1 day. And the biotechnology industry in India is valued at nearly $11 billion in 2016, and next in 4 to 5 years, the value is forecasted at around $100 billion. If you ask me, what is the economics of our own biodiversity? Well, it has much more potential. Like the case of Ongol bull, the farmers are selling at 3 to 5 lakhs, but in American continent, the Ongol bull is sold at 100 to 200, 1 to 2 crores per Ongol bull. And it is estimated that every year Brazilians make around 20,000 crores to 30,000 crores only by Ongol bulls. Imagine that amount of money. You can run a state like Kerala, a state like Goa. I mean, if you have that much amount of money, 30,000 crores they are generating only with Ongol bull. My God, look at my country, see how much biodiverse we are. Why we are not realizing this? Why? Because you see where we are focusing right now. We are focusing right now on technologies that may not be like have great, that great protection towards biodiversity. They may not guarantee that safeguard. See the relevant departments under DSI, Ministry of Science and Technology, spending thousands of crores, hundreds of crores on DSI, established pharma and agricultural livestock institutions, organizations. There are more than 500 startup biotech tech companies in Hyderabad itself who are focusing on this particular technology because there is a lot of value. And who is giving that value? we ourselves, right? So when there is value, then focus will be on one side. But we have to learn from Brazilians. We have to learn from them to realize the potential that our biodiversity can have. So they taught us the lesson, and we have to learn that. But who is they here? I mean, even like at the top, they have to learn. That's what my point is. Now, well, regarding DSI, I have shared till now. So what is DSI? That once I have that genomic sequence, the genetic material with me, I don't have to approach the providers of the genetic resources again and again. You see why it's a problem at the CBD. You know, presently at the CBD, lot of debates are happening on DSI. Many countries do not address DSI in their legislative, administrative, or policy ABS measures. ABS is access on benefit sharing. India requires benefit sharing for DSI, although the requirement is currently determined on a case-by-case basis. Well, what India is doing is, why India is doing this last paragraph, if you read, there is electronic EBS monitoring system currently being developed by experts associated with ABS capacity development initiative, which is under the support of German government. GIS is nothing but German government. Now, what is this ABS monitoring system? It's a machine learning tool connected to several databases containing information on biological resources of Indian origin. So this has been developed by India, which is a good move, but we still not sure like how we are going to progress in it. We have to see because we have so many engineers and software guys available in our country. Now, why India is developing this? Sorry, I think I just want to give example on this. Why India is developing that? Are we taking DSI seriously or not? What happened in this case? Plasmodium vivax. I mean, this Plasmodium vivax and Oxford vaccine patent application. If you are proud of Oxford, well, this is not the good time, but yeah, I think Oxford applied for a patent for Plasmodium vivax sample. The invention means the Oxford University is saying that the invention describes the use of two known proteins derived from the nucleotide sequence of Plasmodium vivax strains originating from different countries, including India. What they did was they collected the Plasmodium vivax sample from Indian patients. That was in the application. Now the National Biodiversity Authority observed this particular thing and they directed the Indian Patent Office to stop further processing of the application. That is, they approached the Indian Patent Office and told the Oxford University to investigate on this case under the Biodiversity Act. Following this, interestingly, what Oxford University did was they amended they deleted all the references of the Indian stream. I mean, all the Indian patient names, etc. They have deleted it from there. Now, according to the applicant, what they are saying is, it's interesting, read it. According to the applicant, the nucleotide sequence of the Indian strain was obtained from a 2005 journal publication. The statement that I read right now is applicable to digital sequence information. Means they got from some journal, the genetic material, the nucleotide sequence is there somewhere stored in the journal, and they got from there. And this is where India acted again. Yes, they were serious about DSI. They told that because we are considering DSI is already part of our law. So you cannot use Plasmodium vivax. And I think it's still the case is going on. So India is considering DSI seriously in terms of benefit sharing mechanism. I can say with some of the case studies at least, and some of the negotiations that I follow. At the same time, with the availability of huge data publicly, when I say huge data, look at your own country, look at your own region, and the endemic things that you have, which specimens are considered as DSI or not is a big challenge, my dear friends. Due to the advancement of technology, DSI is seen as a potential way of implementing Nagoya Protocol. Yes, we can implement Nagoya Protocol using DSI because India is arguing with all the developed countries that DSI is part of Indian law and Nagoya Protocol. So there is no question about it that DSI you cannot like take out, is not under the Nagoya Protocol. Well, this is very interesting. Why there is a debate about digital sequence information? Read this particular, this is a law that EU has developed. The EU guidance document explains that users, that is pharma companies, agrobiotech companies, etc., should respect any conditions agreed upon in mutually agreed terms, including those that deal with DSI. Look at the term they use, should respect. They are not saying should obey or they will be punished, etc. They are saying should respect when they deal with DSI. This is their document, and look at the below paragraph. It's so funny. Just understand in terms of a story, okay? If many of you are so new to this. In this way, the compliance measures are only indirectly relevant for DSI. Compliance with mutually agreed term conditions is not regarded by the EU. Means when I sign an agreement with you, my country is saying that no, it's not my concern. It's between both of you. Okay, this is what EU is telling. Compliance with MAT conditions is not regarded by the EU as bringing DSI within the scope of the EU compliance measures, meaning that there are no measures in the EU regulation relating to the use of DSI. When accessed from a database, that's what I was trying to tell you about Oxford University. When accessed from a database without access to the underlying physical genetic resources. Ladies and gentlemen, this is the classic example. Why Oxford University did all these things? Because the rule, they don't have this specific rule. Yeah, of course, Oxford falls different, but EU, UK was part of EU, in the past, not like after the Brexit, but they had some of the same procedures. But I'm giving the example of EU. So it means that when you can't physically access genetic resources, you can do whatever you want. We are not bothered. So this is the concern. This is the debate happening right now for digital sequence information. I hope that I think I make you understand about DSI in simple language. Well, what other countries are doing? Yeah, Panama is doing really great. Panama doesn't allow its genetic information to be uploaded anywhere in the digital media. If you try to upload, then the Panama authorities are behind you. You can be anywhere in the world. Now, EU wants, now I think this is something EU is, I somehow feel it, okay, fine, not very terrible. European Union wants to implement the DSI using the blockchain technology, which in the third point I mentioned, Ecuador is already doing that using blockchain technology. That's why I tell always that if you want to conserve biodiversity, you have to understand technology. You have to understand the mindset of people who are in this field, like technical field. What is the new technology that is coming? Because with technology, there will be always a challenge for conserving biodiversity. Now, EU in all its statements are clearly saying blockchain technology. What is blockchain technology? Yeah, it's very simple. I'm trying to store a specific information, a specific coding in a block and trying to lock it so that no one can access. And that blockchain belongs to a specific person. So whatever benefits that I get from blockchain that can be transparently shared with the person who is providing me the genetic resources. And all this digital data will be stored in blockchain technology. But you have to ask a question. Blockchain technology is quite new. If you go do trading cryptocurrency, you will understand what I'm trying to say. We never know what's going to happen with blockchain. and there might be hackers. It's still a new thing. And in many countries, blockchain is still like new. There is no law for them. And the other major concern for a country like India or Africa, how you are going to like educate about blockchain technology to the people at the ground level. I mean, with this amount of information, these are some challenges. I mean, it's not possible, but these are like, you have to like work hard a lot. So, I mean, I agree to the second point, but again, I see a lot of challenges in it. So there are many countries who are doing so many things for DSI, they are discussing to get into the post 2020, but that's, we will see how it progress. So by the way, DSI is still in discussion. So if you are thinking DSI is already there in, yes, DSI is already there in some countries, but it's not being like completely like implemented. I mean, not like into in the market right now completely. So this is about DSI. Now, I think what is missing? Well, dear friends and colleagues, what is missing right now? Nagoya Protocol clearly states that a prior consent, a consent should be taken from the local communities when you access the genetic resources, when you access biodiversity. But in our law, in Biodiversity Act, there is no word for consent. It's only consultation, which is a very sad thing. So if you read the Biodiversity Act, the CBD protocols, one of the CBD protocols says there should be a consent, but the Biodiversity Act says no, we will do only consultation. We don't take consent from them. So this question we have to ask to the framers of the Biodiversity Act, what they were doing when they framed this act. So this is a major thing. Again, we have to look as youngsters that why, I mean, when you have ratified CBD, why like you haven't used the word consent? Well, in our constitution, there are words like consent we have to take. In other acts, there are words like consent, but why in the Biodiversity Act, there is no word called consent? It's a question that we have to ask to ourselves and also to our framers of the act. There is no access on benefit sharing clearinghouse mechanism in India. This is complementary to Union of Ethical Biotrade. I will explain this second point in a very simple language. Well, you have a lot of money, but you are saying that I am not going to deposit my money in the bank. I will be away from the bank. But, you know, yearly I will show to the authorities whether I'm paying tax or not. You know, I will do that part. So India don't have access on benefit sharing clearinghouse mechanism. When you have clearinghouse mechanism, you have to submit your reports, your implementation part, your legislation, your administration to the CBD. It's like information sharing. You have to share your information very transparently. What happens when you share information transparently? Well, it improves the implementation part, right? It improves the relationship between the users and providers of genetic resources. What is this Union of Ethical Biotrade? If you collect, I mean, if you buy any like shampoo or any like soap, you can see the ingredients name on that, right? This is what it is. You will know as a person, as a farmer, or, you know, as a person of citizen, I will know like exactly what's happening. India, I mean, in many ways, they do submit so many reports to clearinghouse mechanism, but that is to their will. Okay, so when they want, they will do it. It's so like voluntary for them. Well, the third point, yes, this is very, very important. As young voices should be heard and recognized. Like, you see how I am like furious right now, how I'm, because I have worked with, I have seen all these things. So our voices should be heard as strongly as possible. And this is the right time. And we have to collectively raise our voice, because I have shown you the concern. You do your own research of what's happening and what's the terrible things that are happening when we have like sustainable development and all these protocols and all this discussion, but when they don't happen, then you have to raise your voice. You have to show your concern in any way possible. So ladies and gentlemen, who are responsible for conserving biodiversity? I said young voices, yes. But look, I'm showing you in the an island with a lot of biodiversity. But if you find the bridges are nothing but who are safeguarding the biodiversity from unethical practices, from someone coming people. Yes, someone said we people. Yes, we are the people. At the same time, there are like more than 60 sectors in India, like government sectors or institutions or organizations, NGOs, who has to take responsibility collectively. Well, it's not up to Biodiversity Board of India, National Biodiversity Authority of India, or Ministry of Environment and Forest Change to simply act. They are not responsible. You have Ministry of Agriculture, you have Ministry of Department of Biotechnology, you have Ministry of Science and Technology. Well, all these sectors should come together. You have Ministry of Finance, who plays a great role in providing budget, implementing certain schemes in conserving biodiversity and sustainable development. All these sectors should combine together and act together. What's happening is they're all scattered. I mean, including us, we are scattered. We don't know what's happening in like economic sector. We don't know like the every detail about GST, how much tax that we are paying. So I'm just quoting some examples, but this is what's happening right now. It's not just in India. You take all the countries in the world. It's very chaotic. So I don't know the answer for it. I think we have to like systematically think, and we have to raise our voice, and we have to like make a document where all the sectors somehow should work. It's quite impossible, I can say that, but we can manage that if we like continuously raise our voice. I think this is a famous dialogue from the same movie Jurassic Park. I picked the quote. Dr. Alan Grant, well, says, your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should. Understand what he's trying to say. Pick the word they here. Now, the scientist from the lab says that we are not doing any mad science over here, okay? We have always filled gaps in the genomes with the DNA of other animals. And if their genetic code was pure, many of them would like quite different. But they didn't ask for reality. They asked for more teeth. Teeth here is predation. Teeth here is money. Teeth here is ignorance. Teeth here is arrogance. So guys, understand, we can't blame the scientists simply because people who are into this industry, they are doing the business as usual. But at the same time, to collectively look. And with this, I think I now want to go to a slide for you guys again. Sorry. These are from Jurassic. I recommend you to watch that movie from 1992. It's not 2015. Yeah, next sheet, I'll come to you. Give me a second because I think I want to stop. There are like some questions, poll questions for you guys. I'm just trying to like go to our slides again. And these interesting questions, I feel, oh, some of you already filled it, but please wait before you fill it. I mean, I think you already have the link. If the link is there, kindly say yes in the chat so that. Okay, I'm going to share the link with you again. Copy link, and I'm pasting it in the chat box. Give me a second. This is the link, and I think now I'm going to present anyway. Given a choice, which food would you choose from the below two options? Organic food, genetically modified crops. Well, 33, okay, 6 people, 8 people response. You can see the percentage when coming down. percentage coming down for option A. 15 people responded. Okay, get 17%. Okay, I'm glad. Okay, 16 people. 16 percentage. That's great. Make it 5% people. Come on. Come on, people. Defend this thing. Please respond to this question. How many of you want to consume organic food? Oh my God, you want to consume genetically modified crops. Come on, people. Beat the genetically modified crops. Come on, respond. Respond for organic food. I'm actually betting for organic food. Come on. Please don't make the GMOs win this game. Come on, people, respond, please. Go to the link and yeah, 80%. That's great. Come on, make it 5%, make it 10%. Defeat that. Show like all your frustration here. 25 people responded. Great. Yeah, I think there is no correct answer for it. There is no correct answer for it. It's your opinion. I think the question is given a choice, which food you would choose from the below two options. This, I think that's great. You would like to consume organic food. That's great. 26 people responded. Great. Thank you, Sharanya. Now I will just like to move to the next. I think I explained to you about digital sequence information. According to your opinion, do you think there should be a strong monitoring on DSI? There should be a strong monitoring on DSI. The question is, digital sequence information should have strong monitoring. So what do you think? Yes, it must be. Yes, urgently, definitely, great. Surely, yes. Think if there is no, yes, it must be. Yes, definitely. Yes, it should be. Yes, very strong monitoring. Definitely. Yes. Compulsory. Great. Compulsory. Yeah. Very strong. Immediately. Yeah. Needed. Come on. Fill your responses with this. Very, very important. Very strong monitoring. That's great. Yes. Yes, it should be. It must be, definitely. Yes, I'm very glad that you understood something about it. Something about digital sequence information. Mandatory. Yeah, it should be mandatory. Well, undoubtedly. Okay. Important. Great, people. Great. I think you understood the core thing about at least the important aspect of DSI. Well, I have to tell you, I love DSI, but as Ankur said in his presentation, if it is followed unethically, it can be like, we never know. Right now, yes, it's needed. It should be compulsory. Yeah. I think I'll move to the next question. Thank you. We got like great responses, more than like, I think 43 people responding to this. I mean, that's great. I think I will move to the next question. The next question is on, with regards to access on benefit sharing mechanism, states should take consent from the local communities. State can take consultation rather than consent. State can take decision without consent and consultation from the local communities. What do you think? Great. Okay, people are responding. State should take consent. 83% of the people. State can take decision without consent. Oh, okay. 71% is responded that state should take consent. Okay, 66%, 70% again. Yeah, I think your opinion matters in this. It's just your opinion. Okay, state can take consultation rather than consent. Okay. Okay, I think 18 people responded till now. Let's see for like some more time how many people will respond. Okay, thank you so much for this. I think 66%, it's a good percentage that, yeah, thank you, Sumit, thank you. How different sectors, departments, institutions can come together to conserve biodiversity? I mean, how you think, what do you think about this? I'm saying awareness. Okay, yeah, awareness is quite important. Awareness. Any other responses? Yes. Build up. Okay. Okay. Come together. Be active. Yes. Great. Yes. Build up. Come together. Collaboration of work. Common thought. Yeah, which is quite important. Collaborate. Yeah. Awareness. Great. Great. Yeah, Sharanya. I think wait for another 5 minutes. We will like give you a chance to all of to ask questions. More awareness, more awareness. Okay. Thank you so much for this. I think it shows that you understood. Okay. Now this is very interesting. Which place comes to your mind when I say Biryani and Ali? I am not one response. Yes, Hyderabad. But I'm asking this rice farms. Okay, fine. But biryani halim is made from wheat and mutton. But I got you. I think it's for biryani you mentioned. Okay. But that's interesting answer. Rice farms. Mutton. Okay. Farms. Grocery shop for spices. Hyderabad. Hyderabad. I love halim. Okay. Any more responses? That's interesting. Chicken. Any interesting Delhi street? Wow, I don't know that. But in good, I mean, thanks for that response. Aroma of spices. Yeah. What is that? B Mart? Biryani Mart? You mean? Hyderabadi. Hyderabad. Poultry for chicken. Okay. I see non-veg. Yeah. Farm for wheat. Oh my God, when I say biryani and halim, 45 people responded for this. 46. You people, there are only 51 people in this. India. Great. Yeah. India. Okay. I got you. Pista house. Great. Pista house for halim. I have to tell you, halim got the GI tag. GI tag is geographical indication tag. The first non-vegetarian food that got geographical indication tag is Hyderabadi halim. So 56 responses. I mean that some people should have responded twice. Thank you for this. Oh, sorry. Okay, I think I'm not sure why it's like this. But yeah, I think I stopped the screen sharing. And I think I will take another 5 minutes because this is very interesting for you guys to understand something about endemism that I was actually referring to. Well, I think why I asked that question about Halim and Hyderabad, as you know, it's something related to endemism. Endemism means something very native, something native to a specific geographic location. They are not like everywhere. And so that's why conservation of them becomes so important, and identifying endemic plants and animals is quite important. So now we will play a game. Okay. Yeah, You can type your answers in the chat box. We will see like who is responding. Hello there. I am a fowl having black flesh. I am originated from the Jammu and Dhar districts. Well, I can see some of them already responding. I am also available in Bastar district of Chhattisgarh and Gujarat. In 2018, the Indian government gave me the GI tag. Yes, as some of them already responded, it's Kadaknath chicken. Yes, Kadaknath, you're right. Kadaknath chicken is generally grown in Jammu and Dhar districts of originated from MP, which is endemic to that area. And yeah, I'm Kadaknath. Okay, I next move to the next question. Hello, my name is Chang Tangi. I'm a domesticated goat and are raised by nomadic communities called the Champa in the Changtang region. I provide the world's finest wool and the famous Pashmina shawls. Can you guess I'm found in which place in India? Look at the goat and try to guess. I got you, Sharanya. You said Kashmir because of Pashmina. Okay, we are interestingly, everyone is responding Kashmir, Kashmir, Kashmir. Srinagar, okay. Okay, I think the answer for this is Jammu and Kashmir. Yes. Answer is Ladakh, ladies and gentlemen. If you have heard about the Nuri goat, which was the first cloned goat, I think in India, you can search about Nuri. It got cloned from its embryos. I think it should be so easy for some of you. I'm a shrub found in Shola forest of Western Ghats. Nilgiri Hills got its name because of purplish and bluish appearance of my flower. I blossom for every 12 years. Last time I blossomed in 2018. Do you know my name? Do you know the name of this plant? Can you guess? Don't do Google search. I give you like 15 seconds of time. Yes, you are right. Kurunji, Neela Kurunji. Neela means blue. Nilgiri Hills got its name because of the flowers that blossom in this color. Yes, you are right, Jonathan. It's Strobilanthes. Neela Kurunji, Strobilanthes puntiana. I quickly move to the next question. We are running out of time. That's why I'm moving quickly. Well, let's see who will answer this. My name is Sangai, also known as Dancing Deer. I'm found in the only floating marshy grasslands national park in the world called Kaibul Lamjau National Park. It's part of Loktak Lake. Yes. Can you guess I'm endemic to which state in India? Someone said Manipur. Someone said Manipur. Any more responses? Let's see. Try to think. Someone said Manipur again. Manipur. I guess the right answer for this is Manipur and state animal of Manipur. Yes, you are right. So this is endemic over there in Manipur near Loktak Lake. And let's move to the next question. Well, let's see who will answer this. It's a tough one, but see, I'm categorized as PA. I'm a tiger reserve. I lie close to Eastern Ghats as well as Western Ghats. Thus, I serve as an important biological bridge for the biota of the entire Deccan Plateau. The Suliga tribes are part of my ecosystem, and for the first time in India, the community forest rights have been recognized in protected area and inside a tiger reserve. Can you identify me? Can you identify this place? Let's see who will answer this. No, I think it's okay. It's not. Yeah, I think Karnataka. Okay, Nagarjuna Srisalam Tiger Reserve. Okay, you are saying evergreen forest. Okay, I will see more Karnataka. Someone said Satyamangalam Tiger Reserve. Okay. Sharanya said Karnataka. Yes, you can see the response. It's Bilgiri Ranganatha Swami Hills, BRT Tiger Reserve, Bilgiri Ranganatha Hills. That's the answer. Well, I think with that, I stop the screen sharing. I hope you enjoyed the session. Now I think I can see some questions. If you have, you can ask. Yeah, Sharanya, go on. First you, and then I see Nikshit also raising his hand. So I give first to Sharanya and then to Nikshit. Yes, Sharanya, your question, please.
So the question which you asked about how different sectors or departments or institutes can come together to, in a question you asked, right, so conserve biodiversity. So can you tell the answer for it, sir?
Well, I
So
mean,
could you hear me?
yes, Sharanya, I got your question. So if I understand your question is how come different sectors can come together and conserve biodiversity? Yes, they have to come together. There is no choice if we really care for them. Well, the problem that I can tell you is that we are acting very like separately. You know, each and every department acting separately, and they have their own targets. They have their own targets. They have their own like agendas. So this disconnection should be like, this disconnection should go, and there should be a common connection between them. Um, and for that I think they can organize many forums. The real concern should be raised within all these sectors. What's exactly happening? See, presently all the sectors are acting of COVID. So we act whenever there is a real concern. Unless there is no concern, we kind of, become docile in that, or we generally don't care. The other point is that because when you say sectors within the governments or institutes, uh, the most of the people that are at the higher position, sensitization is very, very, very important. Awareness is there. I mean, I know that there are tigers in India, right? Or there are like lions in India, or there are like some poverty is there in India. But I'm not sensitive to it. I'm not like sensitive. What should I do? Okay, let it go. If I show that type of concern, it's going to be difficult. So how they can come together? I mean, there are many ways actually. If you look, they can come together, but they have to like pick such specific one simple way. And political will plays a great, great thing here. So yeah, I think youngsters like you also should write articles and do research on it, and how we can work together and how these sectors can come together. Yeah, I see Nikshit,
Yes, sir.
and then yeah, Sharanya, go on.
Yes, sir. I have another doubt, sir. What is the digital sequence information on genetic resources?
Can you please repeat your question?
Sir, what is digital sequence information on genetic resources?
Okay,
DSI on
so
genetic resources.
okay, I'll explain to you. Like you grow a neem tree at your backyard, and I come to you and I said that I want the neem tree because I just want to make business out of it by making soaps and etc. etc. But you demand me that you have to share a benefit for selling that soap in the market. I say okay, fine, I will do it. And let's say I buy at 100 rupees per kg and I'm selling at 1,000 rupees. Then I have to pay you a profit sharing at around 10 rupees or 5 rupees, whatever it is. Okay. But I'm so worried. I'm like coming now. Why should I pay you? What I do is I developed a technology called digital sequence information. So what is it? I'm taking the sequence, the genetic sequence of the neem tree, developing everything in my lab and storing the information digitally in my lab, so that I should not come to you again and again. And through that genomic sequence, I can actually make my soaps, my shampoos, and sell in the market. And who is the loser here? You are the loser. So I mean to say that this is what digital sequence information means to make you understand. So you have to think in a larger context. Yeah, yeah, Nikshit, go on with your question.
Yes, sir. Is there any relation between Noyaga and DSI, sir?
Good, very good question. Very good question. The entire debate of CBD is happening. CBD is Convention on Biological Diversity. All the governments right now debating whether we should include DSI in Nagoya or not. Okay, India is telling that, look, all the countries in the world, DSI is part of Nagoya Protocol. DSI is not separate. Whereas some of the developed countries who has advanced technology, especially in biotechnology that we spoke, are telling that DSI is not part of Nagoya. This debate is happening between developed and developing countries. So there is great relation. According to the Indian Biodiversity Act, DSI comes under the Nagoya Protocol, that is access on benefit sharing. You cannot separate DSI.
And so you said
Yeah.
and you
repeat?
said that Nayoga, Mr. X and the mediators, are they having connection, or just in Telugu they say Ankokunda ayindani? They'll say no,
Okay.
sir. Like
Good,
that.
good question again. I got you. I think what he's asking is that, is it coincidence that these people get trapped when like unethical practice happens? I think yes, coincidence is something, it can happen. But at the same time, I will find, let's say I'm doing business, I'm the Mr. X, I'm the Mr. X. What I will do, I'll try to find out who are the guys. I will investigate who are like struggling right now, who are unemployed. I do a lot of my background research and come approach the right person. So yes, I mean, the mediators generally kind of, they may not be completely aware. Sometimes they are aware, but at the same time, you can say that they are our own people sometimes. So I mean, all they are doing is because they need, it's for their own livelihood. Maybe like the cases that I have seen in my, like, while working. Yeah, it happens sometimes, but they get trapped also.
What are the disadvantage of DSI, like failures when they do this?
Unethical practices, for example, I don't share my information, the digital sequence information I don't share. Like I told you in the beginning of the presentation, there are 3 repositories in the world for digital sequence information. One is in United States, second is in Europe, third is Japan. For example, anything like in India, we are we have certain genetic resource, and if the sequence, the digital sequence information is being like manufactured, developed, the genomic sequence of, let's say, a neem tree or a any other specific plant, then what happens is most of the biotech companies, what they do, they store the information. They store the information internationally, and this information is available in the data form. So that's one of the most like disadvantage, I would say. At the same time, once it has, like, you never know who is going to share the information. I have shared the Oxford University case, right? Where, I'm not I'm not concerned. Like, if I have the information, I can do unethically. I can like make my own things at the lab, and I don't share the benefits out of it. So, or the Ongol bull case, the classic case of Ongol bull. Yeah, I think I see Angelina raised her hand.
And
Nikshit, I come to you.
okay.
Yeah, am I audible?
Yes, you are.
So, like, so every day we come across newspaper articles, or even if there are so many animals and plant species that are making their way to the red list. I mean, the endangered and extinct species. So, what if we take the sequence, I mean, the genetic coding of the endangered animals and use them in the digital sequence? So, it is a very possible solution that if we do this, we can create, we can, save those animals. But why are people not taking the initiative? I mean, why are the scientists not taking the initiative on doing this?
Good question, Angelina. I direct this question to Ankush. Ankush, would you like to answer this part?
So right now, like it has just started. They're like starting to sequence each and every organism, like because earlier the cost was too high to sequence, but now like as the cost is reducing, like it now like to get a whole genome sequence, it's costing like $1,000 or less than that. So with reducing cost, it is now possible to sequence every species on the planet, but like it will take time, and there is huge amount of data which comes through the sequencing. Storing that and analyzing that also take time, and lot of junk data is also there. So like analyzing is taking time. That is the main drawback of the whole thing. But like I guess like sooner it will be available. Like most of the species will be sequenced.
Thank you, Ankush. To answer your question, Angelina, well, I think you have to go to United States and see like already some of them already being like being like created using genomic sequences. The example I gave you is Nuri, the sheep from Ladakh, and also the Ongol bulls. You see what happened with Ongol bulls. More than in India, there are like, I think some 4 or 5 crores of Ongol bulls are there in Mexico and Brazil. So yes, I think it is possible. But if I say genomic sequencing, I mean, I love digital sequence information and all these things because I come from that background, but naturally also it can happen. Naturally, many of the plants and animals do like breed. In the wild, we never know what's happening. So yeah, I think I see from Bino, if I am
Yes, Ben.
right in pronouncing your name. Yeah, Bino.
Yeah. So thank you for this opportunity. So I have a doubt. Like due I'm connecting this to the modern wildlife issues. Like so, like if you take the example of illegal wildlife sale and trafficking, if you take that as an example, and their animals are sold dead and in few cases alive. So so when an animal is shipped abroad, let it be through illegally or legally, and it breaks loose, do you think, and also it also breeds in the in the area which it broke out, so it becomes an invasive animal. So if it continues to live over there, do you think like the animal would evolve, or the animals around it would evolve, or would the become extinct?
Yeah, I think that's a good question again. I will answer this in 2 parts. One is domestic and one is wild. See, most of the domestic things are transferred because there isn't some economic value to them. But your question is related to wild animals. So I can give you examples of tigers. So tigers got, I think this happened, everything happened in the past. Tigers got like shifted to United States. Right now in US, there are more tigers in India. So they have their own safaris and mini pockets of safari patches or some zoos, etc. Now regarding invasive species, that's what happening in Australia, where they kind of took a deer called as sambar deer. Sambar deer is one of the like deers from India. You can check about sambar deer, and it is there even in Sri Salem Tiger Reserve and in many of our like wildlife sanctuaries and protected areas from north to south. So sambar deers have been like shifted to Australia many years before. Right now it became an invasive species. Of course, it got adopted, but the problem with sambar deer is that it is kind of eating everything. So it's not allowing the other like natural plants or native things to grow. So they started culling. They do adapt. I think we have example of Parthenium here. Think it again, like there are many classic examples of that. Where when invasive species, when they come, why you are calling them as invasive? First of all, they are not getting extinct. they are actually like breeding. They are multiplying again and again. That's why you are calling them invasive alien species. Yeah, I think most of the cases they do adapt. I'm not sure of the cases where they got extinct. And if they got extinct, that is fine for me. Yeah, Nick,
Thank you very
again,
much.
you raised your hand. Thank you, Vinay. Yeah, Nikshit, go on.
Sir, is there an instrument for those calculating those DSI genetic codes, sir?
That's a very good
For
question.
DSI.
Yes. And when I was thinking about this thing. Yes, I think there are instruments. Nikshit, can you just mute yourself? Yeah, thank you. I think there are many instruments, but I direct this question to Ankush again.
So right now we have three main sequencing platforms. One is from Illumina. So they are the ones who do a lot of genomic sequencing. And then like Oxford has came up with a new sequencing platform called Oxford Nanopore. Those are like mini chips. It will be like size of your finger, and that can do the sequencing, and that you can take it anywhere. You just have to put the DNA on it and it will sequence. And then there is one more company called Pacific Bioscience. So these are like three main companies which are in sequencing. They are they make the machines basically to sequence. Yeah.
Thank you, Ankush. Are there any questions to Ankush? Yeah, Nikshit, you have one.
Is there a chance like maximum percent genetic code is shared by the same of twins? Maximum percentage of shared.
Yeah, I
same.
think that I think I got it. Yes, you are right. You are asking about identical twins, right? So yes, I think identical twins do have a similar DNA. Identical twins do have similar genetic code. They possess very little errors within them. So right now we all are here. 99.99% we are similar, but there is variation of .not one, and that variation is making us different from each other. We are very like universal, but in identical twins, I think it poses same. If I take this opportunity, may I ask Shruti and Sudha if they would love to answer this part? And have you ever come across like in this?
Yeah, I think twins do share. If they're identical twins, then they have the same genetic makeup. But what makes it different is that they'll have mutations which are specific to individual people. Like, for example, if there are two girls for identical twins, then the DNA will be same, but they have mutations which are individual, specific. For example, girl A will have mutations that are specific to her, and girl B will have mutations that are specific to her. So basically, the DNA will be almost same, but the mutations will differ. That is variations.
Yeah. I think I just want to ask this question to Ankush. I think Ankush, I see like there is
Yeah.
lot of unacceptance and lot of ignorance from scientific community, especially from this biotechnology background, and most of them that I know personally, like they're not even aware of the beautiful things around them, and they are always like live in some kind of world where the basics or ground to earth things that are there. So how, even though if you say, I mean, in this scientific community that we are trying to conserve biodiversity, but when you don't understand exactly what's happening, so and only think in terms of economy, so how this scientific community can contribute and what needs to be done?
So yeah, so at multiple levels, scientific community can like sequencing is one of the thing where like scientific community can contribute. And then for conservation, we have the like technologies like gene drive or something with which we can know if there is something you can add to an organism to make it more fit to survive in a new environment, or like modified environment which we are creating right now. So that can help a species to survive. So these are like two different outputs and then we can study how an organism has evolved in a new environment, and that can also be answered by in a research lab basically. Yeah.
Okay, I think that's great. Thank you, Ankush, for that. And I think we will all take group picture. We already passed the time, but thank you for your patience. And we'll take this picture. This will be the last session, and we have the closing ceremony on June 5th. Do join that closing ceremony to like witness like how you all participated and what are the recommendations and everything that we got from all these sessions. So I'm gonna like take all the group picture. I will wait for 5 seconds if others like to switch on their camera. Okay, I'm gonna take the screenshot. Smile, cheese. Got you. Thank you so much, everyone. I think this will be the end of this final session. As you can see, Shruti already shared the feedback form. We appreciate Dr. Ankush's time here, and it's early morning for him over there, but he still joined. And we thank you, Ankush, for your time and for your explanation about gene drives. And I hope that if not everything, at least something we can take. I mean, most of you understood a part of it. Even I also like learned so many things because I have to prepare the presentation. So I read some documents. So I once again thank you all and making this all the entire session so successful, and your opinions and all the things matter a lot to us. So thank you once again. Thank you for joining. I will see you at the closing ceremony. Take care. Bye-bye.
Thank you, Bharat. Bye.
Thank you. God bless you, sir. Bye bye.
Sir, can you please share the link again?
Thank you. God
Did you share it?
Thank you. God bless you, sir.
Sir, can you hear me?
Thank you, sir.